He did his PhD degree from the University of Minnesota in 1992. Subsequently, he was a postdoc at the Minnesota Supercomputer Institute and then joined IISc Chemical Engineering Department in 1993. In fact, that's a couple of years before I graduated. So I know him as a young faculty, joining the center, the institute and going around. So he held visiting positions at the University of North Carolina, James Franck Institute, University of Chicago, Department of Material Science and ETH Zurich. He is a member of the National Academy of Engineers and National Science Academy. His research interest is primarily in the molecular level understanding of structure and dynamics of systems. To be more specific, this includes biomolecules, membrane protein interactions, biomembranes, and many other things. So let us, again welcome Professor Ayappa for the talk today. And I hope that the problem with the sharing screen could be resolved. So let us start.
Okay, thank you so much, Joydeep. I mean, it's really a pleasure to be here and to deliver this Institute Colloquium. I'd like to mention, I think I visited you at SN Bose, maybe was it late 90s, early 2000? I don't remember now.
Yeah.
It's a good. We had a really nice visit, and of course our dear friend Surajit was there then, and I remember spending a lot of time with both of you. And actually, I've written to Joydeep to offer my condolences for Surajit, and that's how we connected, and this invitation followed. So I really appreciate you having me to deliver this lecture. So I've just mailed everything across to you, to the to the admin. So hopefully that should be up anytime soon. So let me maybe get started and just sort of give you a broad introduction to what I'll speak about, and when my slides come up, then I can take over and then show my slides. So the work that I'm going to talk about today is something that we've been doing over the last started over a decade ago, but most of the results have been out from our labs in the last 6 to 7 years, and it's really a collaboration between my colleagues in biology, Professor Sandhya Vishwesharaya, who's in the MRDG, and Prabal Maiti in physics and Joydeep Basu in physics. So we've had a lot of joint students in the past, and we have a very vibrant bioengineering program. So Joydeep, this is something new. It wasn't there, I think, either when Suman was there or you were there. It wasn't there in IISc. But this has been a wonderful program where a lot of the research is being done jointly with the biologists and engineers. And one of the requirements when the program started, rightly so then, was every project should be supervised by a biologist and an engineer, and it cannot be done separately by an engineer or a biologist. So that way people really got together to solve problems, bringing their own levels of expertise into it. So we've been looking at this problem of pore forming proteins, and these pore forming proteins are large molecules expressed by bacteria. So a lot of the bacteria, when they mount an attack, they release these small molecules, and these molecules are water soluble, and typically the size of these molecules can range from 10 kilodaltons up to 30 to 40 kilodaltons. Once these molecules are released, and when they see the target plasma membrane, they bind to it. And once they bind to it, they oligomerize on the surface, they form pores, and basically these water channels penetrate through the cell membrane and essentially kill the cell. There's osmotic imbalance, ions leak out. And in some cases, these pores are used to send in another protein which actually goes and attacks the nucleus of the cell, for example, in the case of the anthrax toxin and so on. So this is the problem that we set out to solve. My lab is largely a molecular dynamics, statistical mechanics lab. We do a lot of MD simulations, both Monte Carlo molecular dynamics simulations, but we've also started doing experiments at some level, and we collaborate very closely with Joydeep Basu and Rahul Roy, my collaborator in chemical engineering. We'll be doing a lot of very interesting experiments to probe the interactions of these proteins on these surfaces. And what is exciting now for us is, for those of us who do simulations, is that a lot of the experimental techniques now using these super resolution methods are starting to probe things at the single molecule level. and I have things to share with you. So hopefully if my slides come up, I can start talking. That's a brief introduction. And I'll wait for my slides because I don't think I can proceed further now without my slides. Has the slides been received?
No, sir, I did not receive yet.
I've mailed it to scolp@bose.res.in.
Do you have access to Scolp account?
No, ma'am. They will send
No,
it to
so
me.
then you should say
Okay.
he has
They
sent
will-
it
Okay,
to
let me
Scolp
try.
account.
I have sent it to scolp@bose.res.in. That's the where I got the invitation from.
No, but
And I've also copied it to Jaideep, so you should have it.
Okay, so I have this thing. Bapi, where do I send it?
Sysadmin, sir.
Sysadmin. Okay, I'm forwarding to sysadmin.
Yes, sir.
Yeah, just check.
Yes, sir. I got one PDF. No,
Yeah,
It's a PPT.
No. That's not.
The file that came to me is a PDF file.
No. That's the sir, poster file. That's the poster file.
Oh, no. I think I forwarded a wrong file. Okay, I'm doing it right away.
Jaideep, it's snbose.pptx.
Yeah. So Bapi, check.
Sir, did you send the mail?
Yeah, I did. Yeah, to
Okay,
sysadmin.
okay, sir. Taking.
So I should tell you a small story while we wait. We had Bill Goddard visit us many years ago, and Prabal was introducing him in our main building. Some of you from IISc will know that the seminars are held in our main faculty hall. And for some reason, the projector didn't work. And it was half an hour after we started, and then I think the entire projection system was quite outdated. Then after they revamped the entire projection system, and he sort of cracked a bunch of jokes while we waited. And
Sir, I got one PPT file.
yes,
I'm sharing it. Verify
yes,
it.
please,
Okay, sir.
please, yes.
Yeah.
Okay, wonderful. Okay, so you can maybe go to a
Yeah,
full
I
screen
guess
mode.
this is the file. Yeah. Can you make it full screen?
The only thing is, yes, yeah. Please, you can just go to. There's some videos they may not play, but that's okay. We'll just proceed. You can make it full screen, and you can just move my slides forward.
Yeah.
Okay.
So make it full screen.
Is it okay, sir?
Yeah, you can go to full screen mode.
No, it's not full screen yet.
Yes, it's fine now.
Yeah. Okay.
So I could actually take control of it, but let's not complicate it. We can move forward now. Please forward it. Next slide, please. Yes. So let me acknowledge my collaborators. As I mentioned, Prabal, Sandhya, Jaideep, Basu, and Rahul Roy, Sachi Gosavi, who we worked with in NCBS, and a lot of students who really drove a lot of the work forward. I think it was really fortunate to have motivated students. Rajat Desikan, who's a PhD student who's left, Pradeep also who's left, who's in Harvard at the moment, Ayush, who's doing a postdoc with in RRI, um, and Amit and Ila just graduated. So I'd like to thank all these students for the work and also funding from the Department of Science and Technology. Next slide, please. Next slide.
Bhakti, next slide. Next slide.
Yeah, so this is what I was mentioning, that these proteins are released in some water soluble form. They bind to the membrane and then they oligomerize, and they insert to form pores. So the monomers can change even from a single protein to as much as 50 in some of the large pore complexes that we see. And the diameters of these pores can be anywhere ranging from 5 to 40 nanometers. And the size of the pore is largely determined by the chemistry of these proteins, and the number of oligomers that they form vary from a different class to class. Next slide, please. So they're broadly classified as alpha and beta PFTs, pore-forming toxins. In the alpha pore-forming toxins, you find that the toxins are rich in alpha helices. And you'll find this on the left-hand side, cytolysin A, which has a large number of alpha helices bundled together. These are released in the water-soluble monomer form. And how they are transported out of the bacterial cell is still a mystery. Are they transported in vesicles, and how they actually reach the target cell is not something that's very well understood. Because as you know, the bacterial cell wall itself is complex, especially these are released by gram-negative bacteria, which have the large lipopolysaccharides and O antigen units on the outer membrane. So how they actually leave the bacterial cell wall is something that's not completely understood. Now, on the right side, there are some examples of beta PFTs, and I'll explain why they're known as alpha and beta. These are rich in beta sheets, and when they insert into the membrane, these insert in the form of beta barrels, which get into the membrane. In the alpha toxins, the membrane inserted parts are essentially rich in alpha helices. So this is the large, broad distinction between these two broad classes of toxins, and we've done a lot of work with the alpha toxins, cytolysin A shown on the left here, and I'll sort of indicate some of the differences between the beta toxins as we go along. Next slide, please. So all my animation is gone, but it's okay. I think it'll speed things up here. So what I've shown on top are the pore complexes. I have the cytolysin A pore complex shown with the membrane as well, and this is a class of alpha toxins. Something seems to have strangely changed here, but anyway, this is a class of alpha toxins, and the membrane inserted part are alpha helices. I had a beta toxin there, but it seems to have strangely vanished. But anyway, so what's the importance of these systems? I mean, a lot of the bacterial pathogens, 25 to 30% of the infections are mediated through pore-forming toxins. And some of the most virulent forms of diseases which we are facing even today, for example, there are a lot of people coming down with pneumonia, Streptococcus pneumoniae, Staph aureus, E. coli, and Mycobacterium tuberculosis has also been implicated in mediating a lot of its phagosomal escape through these pore-forming proteins. So these are interesting from very interesting from a disease point of view. When we started looking at it, looking at the problem, there were several aspects of it which we wanted to look at. One was when these molecules actually get onto the membrane, what is the kinetics and pathways for assembly of these molecules, and how do they form pores? Can we correlate pore formation with cholesterol organization? There's a lot of MD work that we've done more recently, which actually can beautifully shows this whole process of how cholesterol rearranges on the membrane. And a lot of the STED FCS work done in Joydeep's lab shows how this redistribution of cholesterol alters the lipid dynamics, as you would expect. From the biology point of view, when we were talking to Sandhya about this in the early days, one of the themes that was emerging was instead of actually targeting bacteria, as we know, there's a lot of resistant strains of bacteria that have been found out now, especially India and China, where we can just buy antibiotics at will. the idea was now, instead of looking at drug molecules to target the bacteria, can you actually come up with some strategy to intervene this pore formation process or block this pore formation process, and thereby mitigate the virulence of the bacteria? Although we don't have results on that, but that's something which is it's a long, broad goal in this problem. Next slide, please. Please interrupt me if you have any questions. You can press yes. So I'll largely talk about the cytolysin A, which is an alpha toxin. Now, the beta toxins have been studied to a large extent because the beta sheets stabilize the membrane inserted complexes. There's been a lot of crystal structures elucidated for the beta complexes and far less for the alpha toxins. So when we started looking at this, the crystal structure for this cytolysin A, which is expressed by E. coli, had just been published by Nenad Barnes group in ETH Zurich. And we said this may be a good system to start with, and it turned out to be a very exciting problem. And I'll sort of share with you some of the discoveries that we've happened upon over these working on this problem over the last couple of years. So on the left is the monomer, which is again rich in alpha helices. On the right is the protomer, and these assemble. The protomer is indicative of the state in which the protein is once it inserts into the membrane. And if you press the toggle your slide, just forward it. Press the. Can you go forward? Yes. It can keep. Yes, wonderful. So what happens is there's some large structural changes that takes place. One is, if you look at the green circle here on the left on the monomer, I'll go a little slow, because I'm not able to move my slides, but I think this would be a good format. There's a region here which is known as the beta tongue, and this is what samples the membrane initially. And this beta tongue undergoes a conformational change as shown in the green arrow into this little alpha helix, which actually pushes into the membrane. Now, as this transition takes place, the blue alpha A1 helix swings out of this 4-5 helix bundle that you see on top. And the alpha A1 helix peeled out inserts into the membrane, as you see in the membrane complex, not very clear, but you see the blue helix in the membrane. And so this is one of the largest conformational transitions that you see among this class of pore-forming toxins. And yes, it would be very nice to get the free energy of these transformations. That's something we're actually working on, although I don't have any results to show today on that part. Go forward. Yes. So this is the the line diagram of the various helices that you see here. And in the middle, the green is the beta tongue, which undergoes a large conformational change. This is in the monomer. And you'll find a lot of the intervening loops also forming helices once it inserts. So there's a large conformational change en route to pore formation. And what drives this conformational change is something that we've looked at. And I'll share you some of our results in that direction. Next slide, please. So these are the broad paradigms for the pore formation. What we find is that the monomer on the left undergoes a conformational change and inserts into the membrane. And the assembly can take place while the protein is inserted, right? And this is known as a growing pore model. And it will form various oligomeric intermediates, which might have 1 to X number of mers. Typically, this molecule forms a 12 mer complex in the pore state. And these membrane inserted states could potentially be lytic in the sense that they could form water channels and also cause a disruption even before a full pore is formed. Alternately, if you look at the bottom cartoon, which is known as the pre-pore model, these would assemble on the membrane before it forms a pore, and then it would insert to give you a pore where the lysis would then take place. So which of these two models are at play? That is unknown, and that's we still don't understand it, but I'll show you some of the results which give us some clues as to which of these models could be at play. The other aspect is from more microscopic kinetics phenomena. What is the assembly pathway? Is it sequential, as shown in the little black dots on the left side, where a monomer goes into a dimer and so on? or is it non-sequential where you have several integer combinations which can form the pore complex? So can we distinguish between these intermediates? Can we actually find out which intermediates are at play which can cause lysis? And finally, the broader question is, can we distinguish between the oligomerization pathways? Next slide, please. So this is a broad overview. As I said, our lab is really a molecular dynamics lab working at various scales, all atom MD simulations, and a lot of mesoscale simulations that we've done in the past, either with DPD, more recently Martini-based simulations, and so on. And we also, I think the exciting part of the work was our collaborations with the Joydeep and with Sandhya, where Sandhya in her lab, they start to do experiments where you actually take take the cells, expose it to the toxin, and then you look for this lytic activity, as you can see in the little vials there, showing that the color changing, where you have erythrocytes leaking as you keep adding more and more toxin. And based on the color change, you can actually get some details about the kinetics of this whole process. And at the right-hand side are the single molecule kind of studies that I was talking to you about. A lot of the membrane disruption events, a lot of the changes to the lipid dynamics now can be measured. Of course, you could measure them using FCS and FRAP. You can also do very interesting FRET-based studies to look at the changes in the conformation of the protein on the membrane. I'm not going to show you some of this work, but our student Ilanila, who's just graduated, does some very exciting work where you can actually start to look at changes in the protein structure and then correlate it with the lipid dynamics. The stead that we had, was typically you can with the new steads you can go down to 40 to 50 nanometers resolution, and that's the length scales at which you can start to sample things if you use coarse-grained molecular dynamic simulations. So what I'll present to you today is this multi-scale view of this whole process of protein action on the membranes. Next slide, please. Next slide, please. So this is something most of you would know, but I'll just sort of run you by it. So we largely do molecular dynamic simulations where we're solving, the classic Newton's second law in various avatars, in various thermostats, and so on and so forth. Go forward. Forward, please. You can go forward again. So this is just to give you an overview. When we start doing the MD simulations, we need high resolution structural data. On the left-hand side, you typically get this from more recently cryo-EM, but in the past, a lot of these structures were X-ray based structures. And interestingly, all the structures actually are found without the presence of the surfactant, presence of the membrane, and they're all done in presence of surfactant. So there's always this question of whether the structures that are seen in the X-ray are going to be stable structures that would be seen if these pore complexes were inside an actual plasma membrane. On the right-hand side, you have sort of, I would say, intermediate to low resolution dynamical data from FRAP, confocal FCS, and from calcium leakage measurements, which I'll talk to you about. And in between, we have super resolution methods, which gives us this molecular level information. Today, down to 50 to 60 nanometers, will probably get better as we go along. We can also track single molecules, and we try to sort of connect this with the molecular dynamic simulations to interpret some of the structures that we think might be there in the experiments. Next slide, please. So first, as I mentioned, when we obtained the crystal structure for cytolysin A, we first started with looking at the full pore complex and trying to see if this was stable. As I mentioned, the pore crystal structure is elucidated in surfactant, and so one has to see if these structures are indeed stable in a membrane environment. So we can run these now for several microseconds actually, and these were the early days where we were running them up to about a microsecond, and we find that these structures are stable. We track the root mean square deviations and all the other standard indicators to indicate that these systems are stable. Go forward, please. And what you find is when we look at a single protomer that's inserted, you see the green helix that's inserted into the membrane, the purple helix that's transformed and inserted in the membrane. We take these structures from the crystal structure. So we're not looking at any conformational transitions. We're just saying that if a single unit of this pore complex is inserted in the membrane, is this going to be stable, right? So we find that if you look at a single monomer in the membrane, you find it is indeed, it remains in the membrane, but there's a lot of flipping and flopping around. It doesn't come out of the membrane though. But when we start to dimerize it, you find additional stability. Go forward, please. So if you look at the dimer on the left corner, this is where you have two of these protomers sitting together. And if you look at the root mean square fluctuation on the right hand side, the monomer in the membrane has the highest. So this is just the fluctuation of the C alpha residues as a function of the residue number going to 303 here. The largest fluctuations are with the monomer. And the smallest, of course, are with the full pore complex, which is yellow, shown at the bottom. And in between is the dimer. So you see the moment you form a dimer, all the fluctuations are reduced to a large extent. You see these small peaks in the fluctuations, which are due to the presence of the loops in the protein structure. Next slide, please. Yes, so the dimers essentially stabilize, so higher order structures would stabilize these various protein complexes. So we next looked at the insertion free energies using Martini-based models to look at the potential of mean force. Now, recollect that the monomer structure is different, in solution than in the membrane. So we start with the membrane inserted state on the left, on the cartoon on the left bottom, and we slowly pull this out using standard umbrella sampling techniques. And the potential of mean force gives us an insertion free energy of around 40 to 50 kilojoules per mole. Next. So this is useful, just telling us that indeed the single monomer is stable. Next, go on. But as you pull these out, you find loss of helicity in these in the membrane inserted part, indicating that the exposed alpha helices are not stable. And you pick this up on the panels on the right, where you see this destabilization of these helices. Of course, there are issues with Martini as well. I won't go into that. But by and large, what we feel is fairly reliable is that this large insertion free energy of about 40 to 50 kilojoules per mole. Next slide, please. So what we learn from this is that even a single membrane inserted protein, in this case, which we call the protomer, can form water channels. That's what the MD simulations also show us. That you find that on the left hand side, the yellow arrow shows the water channel that can form simply because the membrane inserted part has both a hydrophilic and a hydrophobic region. The hydrophilic face faces the water channel, and the hydrophobic face faces the lipid environment in the membrane. Next slide, please. So what we started to look at was whether we could actually pick up this transition. Now, in an all atom simulation, unless you do some very sophisticated free energy calculations, which we are trying to do using the string method right now. This is a large protein, so we have to break the protein up into small bits and try to see if we can pick up the conformational transitions. So what we looked at here, this is again largely driven by my student Rajat. He went for a meeting in NCBS. It's a nice thing of being in Bangalore and also working in Calcutta, where you have a lot of other science establishments nearby. He went for a meeting there. Of course, I knew Sachi, and they started talking, and he and Sachi's graduate student said, look, let's try to tackle this problem using these structure-based models, which are used widely for protein folding. And the idea here is that you know both the states, that is the state A, which is the monomer here, and state B, which is the membrane inserted part. And the idea is, can we go from one state to the other? So you actually, in some sense, build in the structure of the initial state, you build in the structure of the final state, and we use what are known as this dual basin potentials, which have dual basins for the dihedrals. So it captures the structure of state 1, which is on the left hand side, and also captures the structure of the state 2, which is on the right hand side. Now, next slide, please. So these simulations are solvent free, They basically only contain the C alpha atoms, and all the interactions are based on the contact maps of the C alpha atoms for the given structure. So let me walk you through what we see here. Excuse me, we also had to code in, develop a small model for the membrane, and the membrane is made again of van der Waals beads, so to say. And for the students here, these interactions are typically of order 1 kT. So the landscape, the potential energy landscape that the molecule sees are very, very low. So it surmounts barriers very easily, and which is essentially the purpose of the method. Now, you see here the monomer sitting, which is termed as M here, sitting in near the membrane. And you run these simulations. I think we ran this for about 90 to 100 different repeats. And then you can actually start to see what the statistics are of the entire process, right? So what we found is 80% of the time, the green region here, which is the beta tongue, inserts into the membrane, right? Because that is, if you look at the structure a little more in detail, the beta tongue is actually buried in, because the solvent exposed parts are buried into the protein. But the moment it sees the membrane, the solvent exposed parts come out, inserts into the membrane, and has a conformational transition. And then you find these membrane parallel configurations known as IM2. But in 20% of the pathway, if you go to the bottom arrow, you find that the blue helix, that's the alpha helix, which actually finally ends up in the membrane, starts to sample the membrane as well. But then this intermediate state IM2 occurs regardless of whether you go through pathway 1 on top or pathway 2 below. Pathway 1 is more favored. Anyway, you end up with IM2. Then there's some conformational changes where the entire protein now inserts, the blue helix now inserts into the membrane. And there's some structural changes. It reorganizes, and you find finally the protomeric state as shown here. Go forward, please. This is one presentation where the administrator can't switch off. I'm sorry about that. Anyway, so what we do is we can sample the ratio of insertion times, and we find that the time taken for the alpha helix to fully insert is much, much slower. And that's what we see in this little histogram on the right, as just quantifying this propensity for the beta helix, that's the green part, to insert. Go forward, please. Forward, please. So you can define order parameters based on the contact maps to define the monomeric state that is on the left and the protomeric state that's a membrane inserted state. So you find that without a membrane, we do not see this transition. But when the presence of a membrane, which is the second panel, the colored panel map on the bottom, the membrane, the monomer can start transiting into the protomer. And if you follow the arrow over a long enough simulation time, it fully converts into the protomer, as it should, because you've actually coded in both the initial and the final states. So this gives us a lot of information as to how this entire transformation could take place. Now, when you look at cartoons of the whole pore forming process, everything is sort of standing upright. They sort of walk on the membrane and they insert. But the actual stochastics of the process is a lot more complex, and these simulations start to give you a feel for what could possibly be the various steps in the pore formation process. And it also allows you to connect with FRET experiments which were done earlier, and we were able to show from these simulations that an intermediate known as the molten globule intermediate, which was claimed to be present in FRET experiments, actually doesn't occur. And you can actually look for FRET pairs which indicate that the FRET assignments in the experiments is non-unique to that particular structure. So you're able to actually glean a lot of nice mechanistic information from these simulations. Forward, please. Next slide. So I don't. Can you play the movie for me, please, if it's there? If you just click on the bar, yes. If you just click on the left side on the
sir.
The dark panel.
No, sir. There is no movie.
Okay, you won't be able to see that. Okay, let's not worry about it. This is just a movie to show you how this transition takes place. So we'll go forward. Let's go forward. So what I'll show you now is switch gears a little bit. So we've looked at how the single monomer can interact with the membrane, and actually we've looked at some of the processes that might take place when we looked at the MD simulations of these structures. What was not known for this particular toxin is the role of cholesterol. Now, a lot of these pore forming toxins require cholesterol for their pore formation. And one such class or family of molecules shown on the left are known as cholesterol dependent cytolysis. So if there's no cholesterol in your membrane, then it's not going to form a pore. This is very well known. And there are in a lot of biochemical assays and studies where they've identified the cholesterol binding sites, which are shown on the bottom left of where the arrow is pointed. These undercapeptide loops which actually insert into the membrane. Go forward, please. So this is Listeria lysine, which is a much larger protein. It forms much larger pores of 30 to 40 nanometers, and we've recently done a lot of work with this particular pore forming toxin. This is very interesting. This is an MD simulation that we recently did of 4 of these protomers in the membrane, and on the right hand side, you beautifully pick up this cholesterol binding site. So you can see all the small gray. I'm sorry, this is a little small. The small gray protein cholesterol molecules, bound to the region near the head groups, which is in brown, sitting near the protein part which likes cholesterol. So the force fields are really quite accurate these days. You actually pick up a lot of these very detailed interactions which have been observed in experiments. And on the left hand side, you will see some lipid molecules which are twisted around, forming what is known as a toroid. And I'll show you more detailed graphical images of that in some of the MD simulations that I'll show you shortly. Forward, please. So what is not known was the role of cholesterol for cytolysin A. There is no specific cholesterol binding motif that was identified. And while we were working on the Go models, our students actually noticed what is known as a cholesterol recognition motif, which is purported to be strong sites or certain groups of residues which would bind cholesterol, and that is actually located in this N terminus which swings out, shown in the dotted purple arrow here. They identified a crack motif there. So there was some indication that, look, these particular proteins might actually benefit from cholesterol, although they do form pores in vesicles and other model membrane systems where there's no cholesterol. Next slide, please. So this is work that we did in collaboration with Rahul. Rahul Roy does a lot of single molecule experiments. So we work a lot with supported bilayer platforms, and you tag the particular protein, and you can basically do single particle tracking measurements, and you can get a lot of information from these experiments. What is more challenging and things that they're doing in the lab now, they're also able to use photo bleaching experiments to look at the oligomeric state. So that's very exciting. And that sort of connects with some of the oligomeric mechanisms that I'll show you, which we predicted using more long-term, long time scale kinetic analysis for the oligomerization process. And of course, you can put cholesterol without cholesterol, and they use turf-based methods to look at the single molecule experiments. So you can actually track lipid diffusivities, and you can do FCS on these systems. Jaideep Basu does a lot of that in his lab. And so that feeds into a lot of our own interpretation from the MD simulations. Next slide, please. So these are routine experiments that are done. If you're not familiar with this, I'll quickly run you through this. So essentially, you take vesicles. They could be giant unilamella or small unilamella, and I'll mention the differences in my next slide. So SUVs are typically in the order of 80 to 90 nanometers. We work with unilamellar vesicles, and you throw these it's in solution, and there's a dye that is encapsulated in the vesicle. In this case, it's calcine. And the moment a pore forms, the dye leaks out. And when the dye leaks out, it starts to fluoresce, and you actually measure that intensity in the experiment. The dye is quenched when it's inside, just an auto quenching process, because when dyes are next to each other, they don't fluoresce. But when they leak out and when they are at low concentrations, they start to fluoresce. So it gives you a wonderful signature of whether the protein that you have actually can form pores. And this is the first step that you would do in the lab. Once you make your protein, you do these erythrocyte lysis or these vesicle leakage experiments to see indeed if your protein is active. And similarly, if you are tagging these proteins to do either FCS or in this case, turf experiments, after you attach the dye onto the molecule, again, you do these experiments to see if the activity of the tagged molecule is being compromised or not. So these are very routine experiments that are done in the lab for these systems. On the right-hand side is a plot of intensity versus time. And there are three different curves here. The bottom one is the blue curve, which is a pure DOPC membrane. These are lipids. I'm sure most of you are familiar with these things, but if you're not, essentially they are two-tailed lipids with a head group and hydrocarbon tails. And then you have in red the systems with cholesterol. That is, you have a DOPC with 30% cholesterol, and the topmost curve is a DOPC with 50% cholesterol. And these are the half-lives that have been shown here. There's a lot of scatter in the data, but nevertheless, what comes out of it is that the half-lives are around 2.8 or 2.9 minutes for the blue, that is in the absence of cholesterol. The moment you add cholesterol, you see a much faster kinetics. So it obviously is indicating that cholesterol is playing a role, although pores can form in the absence of cholesterol, and all our cells anyway have enough cholesterol in them. So clearly, these molecules have evolved to take advantage of cholesterol being in the membrane. So we were able to quantify the role of cholesterol, and that's what I'll share with you next. Please go ahead. So we do single molecule experiments. What you do is you do single particle tracking, and then you can actually track the displacement of the molecule on the surface of the membrane. And what I'm showing you here is the instantaneous square displacement, the two graphs in the middle. On the left is just the protein with the beta tongue inserted and the end terminus sticking out. And on the right hand side fully is the beta tongue inserted and the end terminus swinging into the membrane from the extracellular region. So let's focus on these two, the probability versus the ISD plots. These are very informative. So let me spend some time on them. In one case, there is no cholesterol. And in the second panel, that is D, we have cholesterol. So if you look at the panel where there is no cholesterol present, and if you look at the instantaneous square displacements, what you find on the right hand side is the displacements are higher. So when you start the experiments, so you have a flow experiment, you flow the molecules onto the membrane and you start imaging. So what you find is after it's starting from 100 milliseconds, that is the red, you'll find the one single peak denoted as M on the right, which is shifted to the right. And you wait for about 30 minutes, the black, the light gray curve, you find that the distribution now has two peaks. There's one indicating a fast moving species on the right hand side, and the peak on the left indicates a slower moving species. Right, so you're seeing some shift from a faster moving species. Possibly these are binding to the membrane and slowing down because of some conformational change that has taken place in the membrane. But this is a long time to wait because typically the conformational changes take place within hundreds of milliseconds to seconds. But when you add cholesterol, the panel on the right, you see something very interesting. So let's follow this. At 100 milliseconds, you have the red curve, right? And if you wait for about 5 minutes, the entire red curve has left shifted, indicating that the square displacements have decreased and the mobility of the molecules is far, far less. So the purple curve essentially represents a subset or a population that's very slow moving. The red curve represents a population that is fast moving. So clearly in the presence of cholesterol, you've shifted from these rapidly moving species to something that's extremely slow on the membrane. And when you start to just looking at the data, you start to say, look, possibly in the first case, this beta tongue is inserted, and in the second case, you have a situation where the end terminus is also swinging into the membrane and slowing it down. Now, these are proteins have a large extracellular part that is on the protein as well. So unlike transmembrane proteins, which are largely embedded in the membrane, this has a large transmembrane part, and it has its own hydrodynamics on the cell surface. Go forward, please. So what do the MD simulations tell us? So we did some fairly extensive MD simulations. All these are run for about a microsecond or more. And we start to sample the location of the cholesterol um, with the in respect to the protein. So the picture on the left here shows a cholesterol molecule, that is the blue molecule here, and the residues which are part of this crack motif, which has been implicated in strong binding for cholesterol, are sort of shown here in different colors. You may not be able to see them very clearly. So what we did is we took a single proteomeric molecule. The membrane inserted part is shown in the center, and looked for cholesterol binding. So you run these, you do the occupation studies, you look at the histograms of where cholesterol binds. And what we find on the right hand side is the heat map. You have the inserted beta tongue, which is shown by the green arrow. And on the extreme part, you show the N terminus, which is inserted in the membrane, which is shown by the purple arrow on the left. And you find, very interestingly, there are certain hotspots for cholesterol specifically in the N terminus. So you see the lysine, the aspartic acid, and the leucine showing higher residence for cholesterol in this region. Now, what I'm not going to show you is there's a very nice mutagenesis studies done where they have done double mutants and single mutants, and you can show how by mutating these residues, you can actually turn off the lytic activity. So sometimes a single mutation, especially the Y27, which we're trying to understand now with more detailed calculations, actually turns off lytic activity completely. Next slide, please. So we then started looking at, we had not worked with cholesterol before. Initially, for some reason, we were looking at membranes in the MD simulations where we had no cholesterol, but we start to add cholesterol in there. And this is the MD simulation on the left of the full pore with cholesterol. And let's focus on the membrane inserted part enlarged on the top right region. And what you see in green, the light green, are the inserted beta tongues. And the cholesterol molecule shown in orange, you'll find them actually beautifully locating themselves inside this beta tongue. So they actually just go in and wedge themselves in between the beta tongues. Now this is after the full membrane has actually assembled. But we've also done these for systems where we have fewer number of protomers. And again, you find that these are very favorable sites for the cholesterol to go in. And you'll notice in the pore region, in the purple region that's below, there are no cholesterol molecules inside, obviously, because now the water channel is formed, cholesterol has no place there. It's starting to look for other regions, and this is a very favorable region for it. Go forward, please. So this is very interesting to see, and this gave us a lot of handle on forward, please. Handle on the dual role played by cholesterol. I've expanded here the cholesterol pocket. You can see the yellow molecule cholesterol sitting between the blue helix and the brown helix. These are the inserted beta tongues, and these just reside there for several hundreds of nanoseconds. And you can even take these out of the pocket in your MD simulation, and they'll re-enter the pocket. And we've done some very recent mutagenesis studies where you can change the side head groups here, and you can increase the propensity for cholesterol or decrease it. And if you look at the cholesterol density maps from the MD simulations, that is the 2D density, the pore region is shown in the center, and the lighter regions are the higher density of cholesterol. You find beautifully the high density regions of cholesterol, along the circumference of the pore, corroborating the snapshot that I showed you on top. Next slide, please. So cholesterol seems to play a dual role here. It starts to by initially helping the single molecule to actually bind to the membrane. And then once the membrane forms, it starts to stabilize the entire pore complex by sitting in these pockets. So what I've shown you up to now is largely information at the single molecule level, how these single monomer basically transforms to protomer. We were also interested, as I mentioned to you earlier, and this is something that we probably started more earlier on in the project before we got a handle on the experiments and the MD simulations for these complex proteins, was to see if we could actually look at the kinetics of this whole pore formation process. And what would happen is, if you look at it from a kinetic phenomena, the monomer in solution binds to the membrane. These are the various steps, and there's a conformational change. I'm just looking at the cartoon on top, where the monomer transforms to a protomer. And then these protomers would assemble on the membrane and insert to form a pore. Now, whether they form a pore en route or at the end, that's something these models would be hard to get through the models. But let me show you what we have with the kinetic analysis. So we've looked at, if you look at B and C, you have different mechanisms, as I mentioned earlier, a reversible sequential mechanism or a reversible non-sequential mechanism. And the question was, can we, from vesicle leakage experiments, glean out some information about the kinetics? Next slide, please. Jyoti, please tell me when you want me to stop. It's already 5, but I should be done in another.
We started somewhat late, so we can go on till 5:15.
5:15. Okay, I should be done before that, but thanks. Okay. That should be good. So again, this is the dye leakage experiments that I spoke about earlier. And what you do is you measure the intensity of the leakage. Now, let me give you some time scales here because this is useful. If I look at the time scale for C is the concentration of the dye. And if I look at the concentration of the dye as a function of time, I can model it by a simple first order efflux process. And you can extract out a time constant by which you have which can be related to the dimensions of the pore. That is, LP is the length of the pore, RV is the radius of the vesicle, DC is the diameter of the pore complex. Oh, sorry, this is the diffusion coefficient of the dye as it leaves the vesicle. RP is the pore radius, and NP is the number of particles, number of pores in the system. So for NP equal to 1, we can just get some numbers out. For the small unilamellar vesicles, which typically range from 30 to 100 nanometers, the time scale for the leakage of the dye, that is the time it takes for the dye to leak out of the vesicle, is 1,000th of a second. So in the order of 0.1 or 1 millisecond or so. So the time scale is very, very fast. If I go to giant unilamellar vesicles, then I'm looking at going up to about thousands of seconds, maybe half an hour or so. I'm going into 30, 40 minutes and so on. So when you do these experiments with SUVs, and the data is shown on the right hand top here in red, the black data with the red fit, the time scale here is in the order of minutes. So I'm doing the experiment. I'm measuring the dye. I've added toxin into the solution, and I'm measuring the dye as it leaks out, and I'm measuring the intensity of the dye as it fluoresces, and the time scale is in the order of a few minutes. And the time scale to leak out of a pore is in the order of milliseconds. So what I'm actually measuring, so the moment a pore forms, the dye leaks out very rapidly. In this case, for SUVs, on the order of milliseconds. So what you find here is that this experiment basically gives you the pore oligomerization time, and the kinetics of pore formation is what you're sampling, and not the leakage, because that is a very fast time scale. So you're measuring actually the slower time scale, which is the kinetics of pore formation. So that's the premise of these experiments. So we do these experiments, measure the leakage, and try to fit the leakage kinetics using some kinetic model. We also need to build in a stochastic process. So we assume that this follows a Poisson process, and then we work through this using some stochastics, which I'll gloss over today. But that is also built into the model. But I'll show you part of the kinetics and what the model actually is able to tell us. Yeah, please go ahead. Next slide, please. So what we write is we assume mass action kinetics. Now you can ask whether mass action kinetics is valid for these systems. We don't know the answer to that, but let's try it out anyway. Since we're dealing with small numbers here. So we have a balance for the monomer, which essentially converts from the monomer to a protomer. So this is the first order rate law that we have. And once it assembles and the protomers start to assemble, you can write down the rate equations. They're fairly standard. These we've sort of tried to compress them into condensed form. So it looks like this. But the blue arrow here just shows the rates for Pi by P tau. It just shows the rates of formation of small mers and larger mers. And obviously it's being consumed from the smaller mers and larger mers. So you have a depletion term. So it's largely a balance between formation and depletion. And what you can extract out of various parameters, tau f is a forward time constant, alpha c is a ratio of forward to conformational time constant. And some of these things, the conformational time actually has been measured in experiments, and they say that is the slow step, but these calculations seem to indicate otherwise. So there's some handle on these numbers from the literature as well. And there's a forward to backward time constant alpha b. Next slide, please. So you solve this along with build stochasticity. So this is the experimental data that we have. And if you fit this, you have two time constants. One is a fast time constant, which is around 0.4 minutes, and a longer time constant, which goes into about 5 to 6 minutes. And I'll try to explain. So this gives you a broad feel of the processes that could be taking place, and you can actually start to use these as time constant while you reparameterize and actually fit your data. Next slide, please. So what we have here is, so we tried two mechanisms in the model. We looked at the non-sequential mechanism. So you can do either reversible or irreversible. So in the non-sequential mechanism, if you look at the little cartoon on top, you can have assembly of these proteins from various integers. The question is, if it follows a non-sequential mechanism, our model predicts a given efflux function or the leakage. I've sort of flipped this here from the initial rise. This is a decay. Nevertheless, what we basically to take out of this simulation is that you the prediction from the experiment doesn't match with the experimental efflux function. And also you can, since you're running a simulation, you can get the kinetics, that is the probability of the pores, probability of the different species on the membrane. So P1 will be the single protomer, P2 will be a dimer, trimer, and so on and so forth. So it basically looks like an exponential decay in this case. Go forward, please. And again, if it's non-sequential and irreversible, again, it doesn't fit. You're not able to actually capture the leakage function as we see in experiments. That is the blue line with the which has a lot of noise in it, is the experimental data, and the solid lines are the fit, where we actually assume various kinds of MERs that are formed on the membrane, and to see how it would actually predict the efflux function measured from the experiments. Next slide, please. So next we look at, go forward, please. We look at the sequential mechanisms, where in the sequential mechanism on top in the cartoon, every higher order MER requires the presence of a single MER. That is the protomer. The one single protomer has to be present to bind to, say, let us say P2 to form P3. P3 binds to the single protomer to form P4, and so on. And this again can be reversible and irreversible. The reversible sequential mechanism gives us the best fit to the data. And while running the model, we can look at pore formation from various oligomers in the system. So if you look at 4 to 12 MERs, that's the blue line here, the cyan line, it underestimates the underestimates the efflux function. And if you look at the 5 to 12 MER, it actually matches the efflux function extremely well. Right? And again, you see two regions here. There's a sharp drop in the data. If you look at the red and the blue, and the early time for about a minute, this is where the smaller pores seem to form and cause leakage. And then as you go to longer times, the larger pores assemble. Now remember that SUVs are, this is in the order of, I think we had about 80 to 90 nanometers. There's a high curvature on these systems. So the pores itself are in the order of 10 nanometers, and the diameter is on the order of 80 nanometers. So there's a high curvature being sensed by the proteins. So the propensity to form higher order oligomers might be limited by this high curvature as well. And then if you look at the right hand side, we find the oligomer distribution for this particular set of constants, which gave us the right fit with the experimental data, good match with the experimental data. Go forward, please. Yes, so we can get some handle on the time constants. The forward time constants of 4 seconds, a conformational time of 0.7 seconds, a backward time constant, which is high, but nevertheless present. And the gamma is a ratio there, which I won't talk about. Tau B basically is a backward time constant. Gamma is the protein to lipid ratio, which again is in reasonable match with the from the data that we take from the experiments. And tau b is a backward time constant. So what the key thing here is that since it's reversible, you'll always find a population of 1 MERS. If you look at the histogram on the right, you'll always find a population of 1 MERS because the larger oligomers are releasing the 1 MERS to during the oligomerization process. But if you assume a purely sequential irreversible mechanism, then we do not find 1 MERS being present, as you can see in the histogram. And again, we're not able to fit the data at all. And more recently, from the single molecule photo bleaching experiments, there is a lot of evidence to indicate that we always find 1 MERS present on the membrane, indicating that maybe this mechanism that we're proposing is on the right track. Next slide, please. So the mechanism that evolved was shown in this cartoon here. P1, which is the protomer assembles on the membrane in this sort of partly conformed state. It's not inserted into the membrane though. So these things start to oligomerize on the membrane and they stochastically insert. So I've shown an example of a P4 with the membrane inserted state into the membrane. And we found that typically these lower order oligomers from the model seem to be required to actually predict the data. So indicating that these partially formed pores are playing a big role in the pore formation, at least on these SUV surfaces. Now, these partially formed pores have been observed in AFM images for these larger pore complexes, which have been extensively studied. So you find a distribution of pores on the membrane and not just the fully formed rings, but you find these partially formed arcs as well. So we've seen that, at least from this analysis, it seems to predict that the arcs or these partially formed pores are stable intermediates. Now, this is something we can check using MD simulations. That was very nice, actually, a nice connection that we were able to make. Go forward, please. I'm coming to the end, so we should be able to wrap up soon. So we looked at the systems here, that is the full pore complex. I've shown you the MD simulations as shown on the right. Then we started to look at these intermediates. Go forward, please. As shown in the red circle there in the mechanism. So we did a large number of different MD simulations with different pore states. And what you find here is this is the top view that is shown here. That is 6 MER, 7 MER, all the way to the full pore complex. And this is the side view. So the question is, are these stable in the membrane? And how do the lipids reorient to stabilize these structures? Go forward, please. Go forward, yes. So these are, for example, on the left is a top view of a 6 MER complex and a 9 MER complex. The top graphs are at 0 nanoseconds and the bottom panels are at 50 nanoseconds. So what you find is when we start the simulations, we have lipids that are present everywhere in the membrane, but at the very short time you find the white regions is where the water channels form. So the lipids have been basically pushed out and water channels form. The water channel is stabilized by the hydrophilic parts of the protein on one side. And the lipids, I'll show you soon, reorient in a toroidal conformation to stabilize the water channel on the lipid phase. So clearly this is not a very energetically favorable state, but nevertheless it basically is able to stabilize these partially formed states. And again, on top here I've shown where I've written rapid lipid evacuation for a 7 MER. You find the sequence here where you find from 0 to 50 nanoseconds, the lipids are rapidly pushed out. Forward, please. So again, it's the same thing showing different structures here. So now you can ask, if I have the fully membrane inserted pore 12 MER inserting in, what happens to the lipids? I don't have the images here, but the lipids basically form a micellar structure aggregate and then evacuate from the pore. And we've got some very recent diffusion data from FCS, which shows how the lipid diffusivity is modulated when these pores insert and lipids are released from the system. Although we can't capture lipid release in the FCS, we can actually modulate how the lipid, at least track how the lipid diffusivity changes. So this whole pore formation process starts to change the lipid dynamics in very interesting ways. Next slide, please. So we can look at the displacement by just looking at a survival probability, and which essentially tells you what fraction of lipids remain in the initial region, which are present in this initial region at time 0, and how many lipids are then, how long does it take for this lipid fraction to leave this initial region. So we plotted here a continuous survival probability versus time. So typically what you find is that there is evacuation, is almost complete within 50 to 60 nanoseconds. But interestingly, we fit this to two time scales. One is a fast time scale of 0.45 nanoseconds. The other one is a slow time scale of about 15.97 nanoseconds. So this we think is due to the rapid reorientation is the fast time scale, and then it pushes out, which is the slower time scale, to form the lipid arc that we see. Next, please. So this is again picture of the arcs. It's not the greatest of images, but you start to see in the region which is blue, the water channel. And if you look at the face that is blown up on the right hand side, you find these lipids are in some toroidal conformation, as shown by the reoriented head groups that are now facing the water channel. Next slide, please. And then you can actually measure the hydrogen bonding of these lipids in the green toroidal lipid shown here in the snapshot, and then compare them with the free lipids and so on, and you can get some information about basically what it shows is that the left angle here shows a broader range of tilt angles that these toroidal lipids face, the toroidal lipids, to indicate that indeed these are in a toroidal conformation. And then you can look at the number of hydrogen bonds in these lipids that are now reoriented, and the hydrogen bonding is actually very efficient. So it's able to reorient, and actually one would think that they would be different because of the area per head group now being different, but they seem to pack in a very efficient way to actually give you a similar sort of hydrogen bonding that you would experience with free lipids. Okay, you can see a slight increase. I don't know if this is statistically relevant, but because of the probably the slight increase in area, you're able to see a slight enhancement in the hydrogen bonds. Okay, next slide, please. So then we dug up some earlier micrographs, electron micrographs of cytolysine A. Incidentally, there's a lot of AFM images for lots of other proteins, especially the larger beta toxins, but we never found much data from. Actually, there's no data available, AFM data, for these smaller pores. And we also tried to do this for a while in Jaideep's lab, and with very little success. So it's still an open problem. But these were some electron micrographs that were around. And when we looked at them a little more carefully, we start to see, if you look at the yellow arrow on top, you see the white rings are not complete. You see a lot of white rings that are complete, but the yellow arrows point to rings that are partially formed, indicating that these arcs are present and they are stable, and it sort of is consistent with our whole notion from the MD simulations and the kinetic experiments that these arcs are indeed playing a big role in the lytic pathways, possibly at intermediate concentrations. If you have very high concentration, then there's a larger propensity to form fully formed pores. But at intermediate concentrations, you can form these arcs. And there is evidence from these images as well. Next slide, please. So I'm at the end. I've got just 2 more slides. So what we feel is that cytolysine A can actually form lytic channels with intermediates that are stable, as shown here. In blue, you find the single protomer, and as we keep moving forward, they form these membrane inserted arcs, and you find the lipids now, which form the face that is in the parts of the pore where there's no protein, and the channel is formed by lipids in this toroidal orientation shown in purple here, and that these indeed these arcs are stable intermediates. And the red shows the hydrophilic part and the green shows the hydrophobic part of the protein. Next slide, please. So this is my last slide. So what do we know about the oligomerization pathways? I mean, on top we have this growing pore model where the inserted state starts to assemble and form pores. On top we find this sort of a pre-pore model where the entire oligomerization is forming on the membrane and there's no insertion, and then only when a full pore is formed it inserts. So we think we're somewhere in between. The oligomerization could take place on the membrane, and part of the oligomers form on the membrane. And as the process proceeds, some of these can stochastically insert and form these arc-like states, which are viable intermediates for this process. So, I've sort of given you, I hope I've given you a feel for what we've learned using various methods at different length and time scales to give us a sort of a more multi-scale view of this process of formation. And with that, the next slide, I think that's my last slide. I'd like to please go forward, please. Thank you for your attention, and I'll be happy to answer questions. We have a recent accounts of chemical research where we've actually summarized all this work that we've been doing in this area of the last, I think, close to a decade now. And if you're interested, you could take a look at that. It actually gives you all the experimental and simulation work knitted together. With that, thank you for your attention, and I'm sorry about the hiccup for the team's issue. I've never had this problem before, so Jaydeep, I'm going to blame you. I think some problem at your end. I think we're done. Yeah, thank you.
Okay, so let's thank the speaker, and now it's time for questions.
Hi, maybe I'll start.
Hi, yeah, hi.
Yeah, hi. It was really nice talk, and it's really fascinating that to see this really large scale simulations, and you're really pushing the frontiers of this simulating these large scale systems.
Thank you.
Yeah. So I was curious about from a kind of a thermodynamic point of view, at what stage the protein senses the membrane, like for example the beta tongue that you talked about. So what kind of interactions, so how the sensing goes. So first it senses the interface, which is will be negatively charged, and then the interior is hydrophobic in nature. Then there is cholesterol related effects. So I was just curious that what is the mechanism of the sensing? That can you comment on that?
Yeah, so that's kind of a hard question, because unless you're able to track the, so the initial, it's a buried beta tongue, it's buried in the monomer, and there's a swinging out motion of this beta tongue as it starts to sense the membrane. So obviously it's saying, look, I like this membrane configuration, inserted configuration more than being buried in my monomer state when it actually comes to the membrane. How this swinging out actually takes place, we don't really understand. And we have just, what we have started doing though is, since we know there's this beta state of that beta tongue, we've actually, we did the simulation. We ran the whole thing for about 1.5 microseconds. And what we find is when we put the beta tongue at the membrane interface, right, it cuts into the membrane interface, this starts to form a loop. Okay, and this loop then probably transits into a helix.
So what's the amino acid composition of the beta tongue? Are there some positively charged residues which might do some interaction with the interface, or I mean?
I'll have to look at that in detail. I mean, I could look at it and get back to you, but I don't know the exact composition. But largely, it's beta sheets to start with, and so it's actually buried in a pocket in the monomer, and then it swings out.
It should be more predominantly hydrophobic in nature.
Yes, but then when it does the when it actually swings out and actually forms the alpha helix, then the alpha helix is actually stabilized by hydrophobic interactions in the membrane.
I see.
So it looks like the hydrophobicity is actually helping this transition to form into go from a beta sheet loop state into a helix.
Right. Interesting. And another question I had about the Go model. So I was curious that how the membrane is modeled in this kind of structure-based model. And I was curious that since there is no electrostatics, as far as I know, that then what's the difference between a membrane and just a hydrophobic layer of oil?
Yeah, so, okay, so it's a good question. And I think we cheated a bit there. So the membrane basically is hydrophobic beads. And the beads only move in the lateral. In fact, there is no Go model for a membrane. I mean, there isn't one, but we actually wanted a hydrophobic interface. So the beads are essentially move laterally and they don't move in the Z direction. And then the interactions are tuned. This I didn't mention actually. The interactions are tuned with the beta tongue and the alpha helix to give it sufficient hydrophobicity to insert.
That's interesting.
So in many ways, it's it's a very crude model for a membrane, you know. So
yeah, but it's
we're getting the
interesting to
membrane
see that you got this mechanistic pathways, and
it's actually very interesting. Unfortunately, my movie didn't play, but you know,
sure.
that's supposed to
Yeah,
look
that's
nice.
all from me.
Okay,
Thanks.
thank you. Thank you.
Thank you.
Okay, more questions? More questions?
It's a long enough silence, Jaideep.
Yeah, okay. So I have a question that's related to the toroidal lipid formation, the one which actually you talked to us at the end.
Yes.
So that must be energetically quite costly, right? Because you have to cost several amount of elastic free energy.
Yes.
So where the compensation comes from? Why they become stable? You said there's a long-lived stable configuration, right?
Correct. Yes. So I think, you know, so actually we did that. We looked at the so there are two things. We actually worked out a very crude free energy model for that. But what you see is when the pores form, there's a lot of energetic benefit for the interprotomer interactions, because there's a lot of salt bridging, hydrogen bonding, which takes place in the region outside the pore complex, because that is exposed to water, right? And you have the region inside the pore complex, which has a fairly complex set of hydrophobic interactions, and we've actually profiled these in great detail. I think even to answer Suman's question, there's a paper that we've just recently published last year, which has profiles these interactions between the head groups, between the lipids and the proteins. I'll send that to you. And they're very specific residues which enhance these interactions. But then coming to this, so you have an edge penalty in the protein, right? Because it's not fully formed. So you have it, like an arc, and these edges, there's an edge penalty for it to form. And but that's, I think, largely being compensated by the fact that the interactions between the proteins that are already there are very effective. And so whatever slight disruption you have due to this toroid formation is in some sense being compensated. And we find that the lipid order very rapidly restores itself away from the toroid. So the lipids reorient and regain their most favored sort of configuration in a very small region around the toroid. So there's not much of a big disruption, it seems.
So you're saying those toroid formation is short-lived. I mean, what is the time scale?
No, it's the formation of the toroid. The dynamics of the toroid formation, that is, once this thing inserts, then the lipids are to be pushed out, right, from the toroid. So that time scale is very short. But the toroids themselves are stable for, actually we ran
they regain also, right? I mean, they restructure to form the, you said that they also restructure, right, to form the the stable state.
Yeah,
so what's the lifetime of those toroidal things?
Oh, those, I mean, they, as long as the experiments go, I mean, ours possibly, because so they are stable. I mean, I don't know if they are, obviously the pore is more stable, but these are stable for all practical purposes because they would induce lysis. And I think the time scale you want to look at is how long, the cell has a repair mechanism which starts to kick in typically on the order of 5 to 10 minutes. So if these pores are stable, which they are, I think, at least from the experiments, they're stable for several minutes. They seem to be stable. If these pores are stable for those minutes, then it could be lytic. There's no problem. And in AFM images, they show these, I think they're taken over several hours. So they're able to show these that are stable.
Now, the reason I'm a bit curious about this is the following. See, when you make the toroidal shape and there's a pore, then there's a huge amount of electrostatic repulsion, right? Which is essentially like energy penalty.
Yes.
And the protein part, which actually can get inside, that must be hydrophobic because it has to get into the hydrophobic tail of lipid, right?
Yes. Yes. But it's amphipathic, Jaideep. That's the issue. The part that goes in is amphipathic. Part of it likes to be near the lipids and part of it likes to form the water channel. Right? Okay. So the part of the water channel is formed by this amphipathic face, which is facing the water channel. And the rest of the water channel is formed by these toroidal lipids or lipids in this toroidal conformation. And so, I mean, if you look at the free energy landscape for this, it's all downhill, because these are essentially, evolved to form pores. And so they'll just basically, once they hit the membrane,
but
the entire landscape is
compensation. The energy must be compensated in some way or the other, right? So I'm just trying to understand the compensation.
I think the compensation is largely from just the interactions between the protomers. That must be having that, because, and that's why these evolved to form these large extracellular regions. And there's a lot of, like I said, salt bridging and hydrogen bonding, which goes on in the region outside the pore. So it's a valid point. And it's a good question to ask. My thinking is it's just that. It's just largely driven by this extracellular region which stabilizes the complex.
Okay, so more questions? Okay, if not, let us thank the speaker for this wonderful talk and beautiful insight into a very complicated system. So I shall look forward to get him in person sometime once this kind of situation is over and we can actually meet physically.
I know.
Okay. So thank you so much.
Yes. Boss and Jaideep, just stay on a bit. Yeah, stay on a bit before I leave. I'd like to say hello to you. I'd like to chat with you a bit if you are free. Yeah, thank you. Thank you for having me.
Okay, so the meeting is formally over.